Дорогие друзья, приветствую вас на канале Путь Долгожителя. Сегодня у нас в гостях Лиз Пэриш, руководитель компании BioViva, которая занимается разработкой генных терапий, нацеленных на старение нашего организма. И Лиз Пэриш известна также тем, что несколько лет назад она применила к себе комбинированную генную терапию на свой страх и риск, которая включала в себя ген теломеразы и ген фолиэстатина. Теломераза для замедления клеточного старения, а фолиэстатин замедляет старение репродуктивной системы у женщин, стимулирует рост мышц и снижает уровни системного воспаления. Hello, Liz.
But we met at Sen6 in 2013. And actually, everyone I met from that conference, which makes sense, went on to do something amazing. And by then, you were already a superstar. So it was so fabulous to meet you. And I remember we walked around Cambridge and we did some sightseeing. It was fabulous. I was so happy to meet you.
Yeah. I also remember that conference. You applied gene therapies for the first time for yourself. It has chosen some specific genes, for example, telomerase and follistatin. Why you decided to choose these genes?
Well, in 2013, I was just learning about aging at Sen6, and I literally went from that conference home to start an investment company so that I could find investment for all these fabulous technologies that could not only help an aging population, but could help childhood disease. And I was put in touch with a guy named Bill Andrews, and he had led and spearheaded the search for a telomerase activator in cells. And, you know, this went back historically to scientists who are world famous, who had initially looked at the human chromosome and tried to figure out what was the difference between cells that could only replicate a certain amount of times and cells that could replicate and protect their chromosomes, like our germline cells or the cells that are associated with, children, procreation. So he told me about lengthening telomeres and how that not only could help an aging population, but help children as well. And I got really excited about that. I went looking for funding for his company, and actually funders were not as interested because there was no human data that the gene therapy worked. So I thought, really what needs to happen is a company needs to be built to do these gene therapies, to see if they work, and then based on human evidence, push them forward. Because as you know, 41 million people will die this year of aging-associated diseases. I assumed there were plenty of people who would like to participate who were otherwise dying in a failing medical system. I found a doctor who wanted to move aging technologies ahead as well and wanted to start a company with me. And he actually had a big interest in a gene called follistatin. Follistatin was a gene that he had actually already taken to increase his muscle mass and extend his sports career whilst going to school to be a medical doctor. And he said that, and as we studied, we found that there were synergies between the two genes. One of them, follistatin, helps with metabolic health, and it also seems to help with stem cell signaling, telling stem cells to get active even later in life. The problem is if your stem cells are active, the telomeres will slowly deplete in them, even if they have a small amount of telomerase turned on. So we thought, here's the beneficial to gene therapies. And so in 2015, we started the company with myself taking those 2 gene therapies.
Have you used this gene therapy combination once or several times?
I have used it twice, and I have also taken a small amount of a gene called PGC1 alpha. It's a gene that increases the mitochondria in the cells. It's associated with turning white fat into brown fat and increasing cellular energy, which is a very important thing to do with aging. And then the third one I took was Clotho, and that is associated with, well, geroprotection of anything from cardiovascular disease, chronic kidney disease, liver dysfunction, and it's a cognitive enhancer. And so I'm a big fan of experimenting with these therapies in order to use that data to push towards regulatory approval.
Do you observe biomarkers of aging during therapy or after?
So when we did the first gene therapy in 2015, we were very pressed for aging biomarkers. As you know, the only 2 that really existed outside of blood work were, well, MRIs and telomere length. So the third one that was in its infancy was DNA methylation. And we skipped on that one because there was only the first Horvath paper out. And in order to get that test, it was really cost prohibitive because they were doing them one at a time. You had to go through a university, and we couldn't afford it, honestly. And it had, almost no validation at that point. So those were the 2 markers that we took as pre and post aging markers, which was MRI and telomere length. And then we did blood work, and we looked at C-reactive protein, inflammation markers, things like that.
You're collaborating with a well-known geneticist, outstanding geneticist, George Church, and you did some new experiments published last year in mice. Could you explain a little bit the results?
Yeah, sure. So George Church, Professor George Church, is a co-founder of the company. And he has been kind enough to get behind the mission since the beginning. I actually met him as well at the same conference I met you in 2013. And so what we did is I have taken 4 gene therapies. I have taken AAV with 4 different genes. So for people who don't know, AAV is adeno-associated virus. It is a delivery method that will get genes into the nucleus of cells for long-term and persistent upregulation. So the benefit of viral vectors is, well, number 1, so people are not scared. We make it so they cannot infect you with a viral disease. We take out their payload and we put in therapeutic genes. So they can only deliver therapeutic genes and they cannot replicate. But they all deliver these genes to the nucleus. And so instead of having a drug that you need to take over and over again, it gives you the ability to upregulate a protein for long-term expression. We don't know how long that expression is, but we know that it will be sort of the best set it and forget it for most of the population. So when I took those 4 genes, I had to have 4 different therapies made because the gene therapy delivery method is too small to deliver much genetic material. So this was a problem that we wanted to solve. And we went searching for a bigger delivery method that could get all 4, maybe 5 or 6 genes in it with 1 therapeutic dose. So we went to Rutgers University. We went to a specialized team that worked with a delivery, well, it wasn't a delivery method at the time. We've turned it into a delivery method called CMV, or cytomegalovirus. So in comparison to AAV that can deliver 4.8 kb, which is a size of genetic material, the cytomegalovirus could potentially deliver up to 50 kb. And so that's 10 times the dose. And so the interest when we're looking at treating aging-associated diseases is to look at combinatorial genes. Now, I have to be honest, when we started in 2015, I thought that telomerase reverse transcriptase might reverse all of aging, but it does not. So even though when my data came out, we showed 20 years initially and then even more telomere length gain, meaning that in my telomere length, I may have put on cellular divisions up to maybe 30 some years extension, meaning that instead of dying probably at 78 or 79, I might live 20 years longer than that due to the capacity of my cells to divide. You know, we have to wait and see. That actually didn't reverse all of aging. And so, you know, then we went looking for genes that would be associated with reversing more hallmarks of aging, which, you know, the hallmarks of aging are growing, but some of them are downstream effects of original hallmarks. So anyway, our hope now is to deliver gene therapies that will deliver massive payload. But in the paper, we didn't go that far. Really, we just wanted to make sure that CMV could deliver genes. So we designed 2 different arms that each had 2 arms. One arm was telomerase reverse transcriptase because its action is well known. It had already extended the lifespan of mice, and it was an interest of our company. The second gene we chose, of course, was follistatin because it's also an interest of our company, and the experience in the lab with follistatin is also well known. We chose 2 genes that had been reproduced in multiple labs to see if we could deliver them and get the effects that were also already known, and then see what sort of benefit we could get in the long run. So we basically ordered old mice, and these are wild-type mice. They're 18 months old. They're quite expensive. They are like the equivalent of someone maybe 56 to 63 in human years. And so they're a great candidate for aging-associated gene therapies because they would mimic an age in which we might be allowed by a regulatory body to start taking these type of gene therapies. And when they're proven safe, we'll probably take them much younger. So we treated them 2 ways. We treated them intraperitoneally, which is just an injection into the torso. And then we also sprayed the gene therapy in their nose. Our company had already had interest in intranasal gene therapies. In dementia, we were delivering an injectable gene therapy. With CMV, because the route of infection is through the sinuses, we just wanted to see if you sprayed it in the nose, could you transfect the body? And well, what happened is it did work. It worked as well as the intraperitoneal injection. A couple of the most important factors of the paper is that the gene therapy was redosable. And AAV, you can redose it, but you'll get an immune response. We did not see an immune response. We had over 40% lifespan gain with telomerase reverse transcriptase, and maybe that was the benefit of redosing it over other studies, layering it into the system. So telomerase reverse transcriptase, the enzyme telomerase itself, is only used in the cell that's transfected. So let me tell you the difficulty of that. So if I transfected my hand with gene therapy, let's pretend my hand is my whole organ, it's unlikely that we'd hit every cell in my hand So when I took gene therapy, maybe only my middle finger and my index finger got the gene therapy. The rest of the cells would be aging. So the idea of doing it over and over is to hopefully layer it into, your hand and get, more and more distribution of the therapy. Follistatin is different. It actually shares the proteins systemically, so you really don't have to layer it over and over into the system. But anyway, it was good to try it. So with follistatin, we got over 30% lifespan gain in the mice. And what we saw in both gene therapies were better metabolic effect, a healthier lifespan, looking younger. Follistatin, the mice got very muscular. Telomerase, they maintained their body weight at a better degree than the controls. All the controls died predictably around the time that this is the great thing about mouse studies. They're not very long lived. And when the lab tells you that they're going to live some 28 months, that really is about it. And then the other mice kept going. And, we did not know that we would get gains in lifespan. We didn't even know if we could deliver the genes. So the third arm, of course, outside of the control was just delivering the CMV to make sure it wasn't just the CMV that was causing the lifespan gain, and it wasn't. So that's pretty much the study in a nutshell.
What do you think? How possible to translate this knowledge into clinical studies? Because many regulatory issues exists.
Yeah. So what we did is we went from that study and we started designing the human attenuated CMVs so that people can get up to speed here to see the difficulty in CMV. Unlike AAV, CMV is species specific. Luckily, the human CMV is bigger than the mouse CMV, so that's really good. But it is really quite different. So how would that translate to a human? Well, we hope it will translate really well, and we hope regulatory systems will let us in. What we did to hedge our bets is we started using a CMV that had already been in human studies that was attenuated for a vaccine. And that's where we started. Then we started modifying that. And then follistatin has already been in humans for studies in muscular dystrophy. So when we put together one of our first constructs with a single gene, we used one vehicle known to human studies and a gene known to human studies. And then the hope is that will get us regulatory approval more quickly for the use of that technology. And that would be a nice step in the door. In the meantime, we have constructed a bunch of the CMV vehicles with multiple genes. And so those are things that I'm more excited about taking so that I can predictably get the same genes to transfected cells that I'd like. One thing that we look at, for instance, in cell reprogramming, let's just put, why would you use a bigger vector? So in cell reprogramming, they have a vehicle now, because you don't want to keep the reprogramming factors on permanently. So they have a switch so that you could turn it on and off with doxycycline. Well, with AAV, they have to use two vectors, one with the genes and one with the switch. Well, this has a switch, and then this has the switch control. With you really wouldn't want those to be separate, right? You would want each cell transfected with both, but with AAV, you don't have any way of controlling that. With CMV, we could merge those into one and put them into a cell. We're not working on that right now, but these are the type of partnerships we'd like to have with companies who are.
Many people have immunity against this type of virus. Maybe it could decrease the effectiveness of the therapy. What do you think?
I think that, well, they do to AAV as well, but that's kind of a moot point. Even though people have immunity against CMV, they can't get rid of it. The genes themselves in CMV hide from the immune system. So you can have an active infection your entire life and not know it. So we want to take the benefits of CMV, but make it so it cannot get you sick. So the persistence of the CMV is what we like. On the other hand, AAV, which my company also works with, has immunity and the body is able to silence it. We want a vector that cannot be silenced over 6 to 8 weeks while it transfects the cells and infects the cells with the therapeutic genes. So since all of the vaccines have been failures against CMV, it actually makes it a rather good delivery method.
What do you think? Do we need several application of such longevity vaccine, or once one application is enough?
I think that for some genes, we'll need multiple, and probably even for the long-term persistent gene therapies, you probably will need to be redosed. Outside of integrating genes that will divide with the cell, we're creating what's called episomes. And that's the focus of our company, is to not integrate, to not cause disruption to your natural DNA, but rather to start delivering many artificial chromosomes, right, that code on their own. Now, they're not likely to divide with a cell. So if you have a cell like a neuron, you may have lifetime persistence. If you have a cell like a myofiber, like a muscle cell, it might last for 10 years or more. If you have cells that turn over quickly, like your epidermis, you probably would need more treatments. So one of the ways that we get around that is using genes that share their proteins and then putting them in long-lived cells. For instance, follistatin, a target for it is your muscle cells, because they're long-lived cells and the protein is shared throughout your body, even to your skin. And that could be persistent for a very long time. With telomerase, again, where we only have activation where the cells are transfected with the gene. And so those will probably be redosing gene therapies. Our hope is that with something like an intranasal gene therapy, people would be able to do that at home. You know, maybe a pharmacy would ship it to you. Maybe they would ship you gloves and a system to put over your head at the sink. You would spray, you would take everything off, throw it away, and you would be done for the year. That would be the hope there.
What other longevity genes do you consider promising in the future?
Oh gosh, we look at everything. Even there's some new candidates. Let me see. I've got some new candidates. Let's see, where was I? I was sharing that with a group, but there are new candidates that may even help things like follistatin perform better in cohorts in which find it difficult to get exercise. So what we saw in human studies is metabolic health is better, I think, across the board in everyone who has ever taken follistatin, but it is very dose dependent. But if you're not active, you know, you're not going to gain muscle weight. So you really need to get out there and get active, and that's not possible for everyone. But there's a new gene that we're looking at, which is PCY, T2, and there's several others like it that we watch all of the studies in the gene-based therapy. We also watch studies that have to do with collagen production and things that are ancillary, but also very important to longevity, treating different organs with genes that can help us function and of course look healthier and happier and be happier throughout our lives. I cannot guarantee happiness, I'm sorry, but we're hoping that if you live long enough, you will be happy. I mean, gosh, I have to tell you, this week alone, somewhere I had this open, there are now, there's a group studying gene therapies for autism. There are groups studying gene therapies for schizophrenia. There are companies that are studying, actually, the Belosa disease in children where their skin becomes very fragile. They have now produced a redosable topical gene therapy for patients to do at home. I mean, the whole area is just taking off. And then I was just at the ASGCT, in which is all the research mostly in AAV, a little bit in the non-integrating lentiviruses. And then, of course, what's really popular since COVID is mRNA dosages of both gene therapy or for vaccines. And so for people listening who don't know the difference, gene therapy, a gene is a persistent vehicle if delivered to the nucleus. If it's delivered in some deliveries, it's going to be short acting, like a lot of the lipid nanoparticles that are non-viral based are going to give you kind of a temporary exposure to a gene. But mRNA is a way to dose a gene therapy product with shorter term expression. And the reason that we're not doing that right now is we're choosing genes that seem to be beneficial when persistent. Okay, so for instance, my telomere length from taking telomerase reverse transcriptase in my T lymphocytes went from 6 some kb to 9 some kb 8 years later. So essentially for every year that I age, I actually gain a few years in telomere length. It doesn't go from like short to super long overnight. And so that's, I believe that that's due to the persistence of the gene therapy and that it's necessary.
Thank you so much for your comprehensive explanation for the whole field. And I wish you active health and longevity, eternal youth, and all success.
I wish that for you and for everyone watching, because I know that you have a group of advanced listeners who are actually interested in healthy longevity. And I hope that we can get more people interested. I'm always surprised when people think they want to die later. They never want to die today, but they think that it's okay to die later. I think that when later comes, they won't want to die of disease either. So thank you for all your hard work.